Welcome back everyone to our very first video of 2024. I'd like to wish you guys a happy new year and I hope everything is going quite well. So for those, of course, who are returning, I want to thank you guys for tuning in again. And for those who are new, my name is Andrew Commander and I make educational videos on the CAPS curriculum. So in this video, we will continue on with our life sciences series. So as you can see, we are focusing on meiosis. So this is supposed to be on your week 4, according to, of course, the schedule that your teachers should have. Okay, so let's look at our CAPS document. Remember, this guides us a lot, and so we have to keep on coming here and see if we are doing what we are supposed to be doing. Okay, so meiosis, of course, you should know the definition of meiosis, the purposes, why it's called a reduction division. Okay, the importance of meiosis. Okay, very important that you know the importance. The introduction to genetic variation. Okay, we'll look at all of these and also consequences of abnormal meiosis. Now, for the consequences of abnormal meiosis, I'm thinking that we should maybe create a separate video for this because I really want to break it down for you guys. And I know that a lot of students really struggle with understanding abnormal meiosis. So I'm thinking that's what we should do, create a different video on it. Okay, that's what we are going to do. So in this video, that means that we are basically going to cover just these three. Okay, right. And also, so in the second video, we should look, of course, at these two: the consequences of abnormal meiosis and of course the difference between meiosis and mitosis. Okay, now our previous knowledge, guys, is that we are aware of only mitosis, right? Mitosis. So in mitosis, what really happens, in simple terms, really, you have a single cell. Okay, let's say maybe you were playing soccer whatsoever, and on during the match you get injured. Okay, so you get injured, you got bruised, or maybe there's a scratch on your knee. Okay, so right here on your knee, gotta zoom in there. So let's say you have a scratch somewhere there. So what really happens is that that scratch means that you have damaged cells. Okay, so those cells are damaged, so they need to be replaced. Okay, and we cannot just replace them with any different kind of cells. We have to replace them with identical cells. Okay, so we are replacing the damaged cells with identical cells. Okay, so replacing that, then that means that you will heal. Okay, so that means that you will no longer, of course, have um, that cut there. Okay, so that's what we are doing. So mitosis really helps you with this. Okay, so you have body cells. Okay, so every body cell in you will get replaced if it is damaged. It will be replaced by what? By mitosis, the process of mitosis. Because what mitosis does is that you have this single cell and you end up with the same cell, but we call these daughter cells. Uh, but these two cells have exactly or are exactly identical to the original cell. Okay, so by this we are actually replacing these new cells that were damaged, okay, with the same kind of cells that you have on your knee. Okay, so that's the idea with mitosis. So it has nothing to do with the production of sex cells like your sperm cells or your egg cells. It just replaces one of its functions to replace these damaged cells with new identical cells. Okay, so that is the idea there with mitosis. Okay, so now when we look at meiosis, guys, okay, and also by the way, we also have, so if you look at these cells are what we call somatic cells. I'm sure you are aware of this. Somatic cells. So these are simply body cells. Okay, so that's just these are body cells, and really they have we say that they are diploid. Okay, we will look at all these terms, all the terminology and stuff. You have to know all of that. But with this, just remember that these are body cells, and in mitosis, we're basically producing the same identical type of somatic cells or body cells. Okay, so that's what is happening. Now, with meiosis, it is also a type of cell division, but this is where we are actually producing sex cells. We are making sex cells. We are making the sperm cell for the males. We are making sperm cells. We are making egg cells for the females. Okay, and so in this way, they will reproduce. Okay, so that is the whole purpose of meiosis. Okay, but we will break down all this definition right here, whereby which says whereby a diploid cell undergoes two cell divisions and divides to form four haploid cells. We will look at the haploid. What does haploid mean? What does diploid mean? Because you have to understand all these terms in order to understand this definition. Okay, we'll get there. Okay, you don't have to worry about that now. So that's what we're saying. So we're saying that meiosis enables us to produce what? Sex cells. Okay, sex cells, we call them gametes, guys. Okay, I need you to remember this term. When we are talking about gametes, we are talking about sex cells. Okay, when we are talking about um, for an example, your somatic cells, so your somatic cells are not sex cells. Okay, so these are not sex cells; they are just body cells. Okay, so looking at this, we are saying that we are producing sex cells, which are gametes, right? And these sex cells, of course, they are different from your body cells. Okay, because of course we'll take a look at that. So where does this occur in the body? Where does meiosis occur in the body? So it actually occurs in testes for the males, okay, and it occurs in ovaries for the females. Okay, so these are animals, of course. So these testes, of course, we are producing the sperm cells, okay, and then in the ovaries we are producing the egg cells. So in each and every one of these organs, okay, in every one of these organs we are actually we are producing, or there's actually meiosis that's occurring. Okay, so these are the sex organs. Okay, now in plants it occurs in antlers and ovules. Okay, so these are also sex organs. So all of these are sex organs, and this is where we are producing these sex cells. Now let's take a look at the cell structure. So in the cell structure, guys, we said that within or inside the nucleus we have a network. Okay, so these are just long threads that are made up, of course, of DNA, which makes up genes. Okay, so these are just structures like long threads, of course, that contain DNA. Now, these long threads, um, they can they can become dense. Okay, so they shorten and become concentrated and make up a structure um, that um, that is made up of these two structures. Okay, I'll just break that down right now. Let me just finish drawing this. There we go. So you have this chromatin network, which then becomes this structure. Okay, so this structure also contains what the chromatin network contains: the DNA, genes, okay, so on and so on. So this structure, as you can see, it looks like it's basically these two um, short threads like this, okay, these two, and then you basically they are tied together by what by this structure right here that we see over here. Okay, so you have these two short structures that are combined by this structure. Okay, so these of course have names, guys. We don't just can't just say structure all the time. So these two short structures, we call them chromatids. Okay, so these are chromatids. Of course, they're just short structures which contain DNA genes, and they are tied together by this structure in the middle, right over here, that we call the centromere. Okay, so we call it the centromere, right? So and then the structure altogether, we then say that it is a chromosome. Okay, sorry about my writing, it's pretty bad today, I don't know. So so this is a complete chromosome. A chromosome is made up of two chromatids that are joined at the center by a centromere. Okay, so these are the structures which result, of course, from the chromatin network. This usually happens after DNA replication. Okay, we'll go into that. So we're saying that after DNA replication, the chromatin network forms these structures, which are called chromosomes. What are chromosomes? And these are essentially, of course, long threads made up of DNA and genes. They have um, single threads, which of course are called the chromatids, which are then joined together by the structure that we call the centromere. Okay, so chromosomes are, as I said, are made up of DNA and also a protein that we call histones. Okay, so chromosomes are what we are going to talk about, like most of the time. Okay, when we are studying this meiosis. So these, you have to know the structure, you have to know each and everything, of course, about the chromosomes. So we're saying that these are made up of DNA and protein. The number of chromosomes in a cell is characteristic of an organism. Now, each organism has a unique number of these structures, these chromosomes. Okay, so for humans, as us humans, we have 46 chromosomes. Okay, in each cell. So in each cell, inside this nucleus, each cell in a human has 46, a total of 46 chromosomes. Okay, and that makes us human. Okay, because there's no other species, no other organism that has the same number a same number of chromosomes as us. Okay, so basically each organism has its unique number of chromosomes. Okay, for us we have 46. Okay, in each cell. Okay, in each which cell in each diploid body cell. Okay, have to be specific there. Okay, in each body cell or in each somatic cell. Okay, so each cell has 46 chromosomes for us humans. And then we say that when a cell has two sets of chromosomes, okay, so chromosomes, they can arrange themselves as partners. Okay, so you have, let's say you have this chromosome. Okay, this is a bad drawing. This chromosome and that chromosome. Okay, and these should look alike, really. But okay, excuse my bad drawing, guys. So these should have the same size. Okay, they must have what we also call, so they arrange themselves in these two. So these two will arrange themselves like that, and they will be close together like that. Okay, so they arrange themselves in twos, right, in groups of two. So these groups of two, what they are similar, of course, with size. They have the same size. They also have Remember, we said that each chromosome is made up of DNA, and DNA, of course, has these portions. Portions of DNA, what do we call them? We call them genes, right? So genes, so you'd find out that these chromosomes would have the same type of gene in each position. Okay, so let's say this position, this gene here codes for eye color. Okay, so and that over there also would would code for eye color. I mean, so maybe that it would code for a red eye color, that would code for blue eye color. So it really depends on the person. So these, you'd find that these are inside the cell, and they are grouped like that, and they have the same type of gene in each position. Now, we don't use the word position when it comes to life sciences, we use the word locus. Okay, so the position of a gene in life sciences is called a locus. Okay, so let me rather rephrase that. These chromosomes, which are called homologous chromosomes, okay, so homologous, so these, what makes them homologous chromosomes is the fact that they are grouped, of course, in pairs, and they have the same size. They have the same. They have the same type of gene. Okay, or the same type. Yes, the same type of gene at the same locus. Okay, at the same locus. Okay, because for other chromosomes, for an example, you'd find that you'd have maybe this would code, let's say, for height, and for the next chromosome, maybe the height, the gene that codes for height is right over here. So as you can see, these are not in the same position, not in the same locus. Okay, so these homologous chromosomes, they are they have the same type of gene at the same locus, and usually you'd have each one of them is inherited from the father and from the mother. So this could be maybe from the father. So you got this chromosome from the father, and this you got it from the mother. Okay, so that's what makes these homologous chromosomes. They're just um, chromosomes which have, of course, the same type of gene at the same locus, usually inherited from the mother and father. Okay, so that's what we are saying here, guys. I don't want to confuse anyone. Okay, so then when a cell has these sets, so each cell, let's say you have a particular cell, it has these it has chromosomes that are grouped in twos like that. Okay, chromosomes that are grouped in twos like that. Then that cell, that whole entire cell, okay, remember these are in the nucleus, so that whole entire cell, we call it a diploid cell, guys. Okay, so we say that it is diploid. Okay, diploid for two, right? So it has these it has two sets of chromosomes, okay, where each chromosome has a homologous partner. So whenever you have, of course, a cell that has two sets of chromosomes, each cell has a homologous partner. Then that cell, the whole cell, you call it a diploid cell. Okay, so that's what we are trying to say there. Okay, diploid cell because it has homologous chromosomes or homologous partners. Okay, each cell has each own homologous partner. Then we say that cell is diploid. But when you have a cell that has only one set of chromosomes, okay, so in this case, we mean that this particular cell, okay, has only single chromosomes arranged like that. Okay, single chromosomes. These do not have a partner. They do not have the homologous partner. So this cell then, we then call it haploid. Haploid cell. Okay, hap, H for half. Okay, so it means that doesn't have each chromosome doesn't have a homologous partner. Okay, so that's what we are saying there. Okay, so know the difference between diploid cells and haploid cells. Okay, it's very important, guys. Okay, know that. Okay, Right. So this means that one thing I forgot is that this means that this cell, the diploid cell, actually has double the number of chromosomes the haploid cell has. Okay. Or let's rather say that haploid cells have half the number of chromosomes that diploid cells have. Okay. All right. Okay. Let's move on. So before meiosis begins, so meiosis we group it into phases, stages, right? And these stages, there's 5 of these stages. Okay. I've even written an acronym here. I use this acronym. I use this acronym a lot, even at school. It's IPMET. Okay, IPMET. That will help you remember, of course, the order or the sequence of events or the sequence of these phases. Okay, just IPMET. That's it. Okay, IPMET. That's I for interphase. That's P for prophase. M for metaphase, A for anaphase, T for telophase. Okay, so we will explore all of those. You have to know each and every single one of them, know the sequence of events, know important events that occur in each and one of them, each and every one of them. And I promise you guys, it's not difficult to really get a hold of these and remember them. And you just have to know the most important events, and you will ace everything. Promise you. So we're saying that before meiosis begins, okay, so we say meiosis begins actually just after the first just after the first phase. Okay, so this is what we consider as meiosis. Okay, the PMET part. Okay, the I part is simply um, a preparation, okay, of meiosis. Okay, so when I say before meiosis occurs, I'm actually referring to this interphase. Okay, we're actually referring to the interphase. So what happens right over here? During the interphase, you still have this chromatin network. Okay, you still have these long threads. They have not turned into these structures that we call chromosomes yet. Okay, they are still chromatin network. That is in interphase one. So what happens then? Okay, so what happens is that DNA replication takes place. Okay, you guys, we went through DNA replication. We know all the steps, what happens. So it's there really to double the genetic material. So that's the purpose of DNA replication. Okay, because what will really happen here for meiosis, unlike the mitosis, where you are going to produce two identical daughter cells, with meiosis, you have this one cell and you end up producing four. Okay, so you end up producing four cells that are not even identical. Okay, these are different from each other. There's four of them though. Okay, so that means that we have to double the DNA there, so that each one of those, of course, will get or will share that DNA among each other like that. Okay, so we have to double it. That's why DNA replication occurs. Remember, DNA replication, we are basically producing a replica of this DNA. Okay, so we are doubling it. Okay, so that's the purpose of DNA replication that occurs. It's actually to prepare this where you are producing four cells from one cell. So it's there to double the genetic material so that it is shared among those many cells. Okay, right. So DNA replication takes place. That's the first thing, and then each chromosome now will consist of two chromatids joined by a centromere. Okay, so after DNA replication occurs, that's when you start to produce, or that's when the chromatin network actually densifies and become their chromosomes, or they form these chromosomes. Okay, so that's what happens. Okay, and remember, this actually occurs in a diploid cell. Okay, so we are taking a diploid cell, a body cell. We are taking a body cell, And from this body cell, we are going to produce 4 different sex cells. Okay, by different, guys, I mean that they have genetic material that is slightly different from each other. Okay, and they are variable. Okay, there's variation that occurs, and we'll look at that as well. So we are taking a diploid cell which has those homologous chromosomes. It undergoes 2 cell divisions, and then it divides to form 4 haploid cells. That means that these cells that is going to form, these cells, each cell will not have homologous chromosomes. It will have chromosomes that, of course, that do not have homologous partners. Okay, and that's what makes sex cells different from somatic cells. Sex cells have half the number of chromosomes as body cells or somatic cells. Okay, so you're taking this diploid cell, then DNA replication occurs, and then you are forming these structures from a chromatin network. Now you have chromosomes. Okay, so as I said, DNA replication is there to help double the genetic material so that it can be shared by the new cells arising from cell division. You have to know this reason, right? Please, guys. That's what DNA replication is there for. Right, so now we have these chromosomes, and each cell, of course, is diploid, right? So we take a diploid cell. So now, there we go. Look at this. Okay. These cells, as you can see, guys, they are arranged in pairs. So this is a homologous These are homologous chromosomes. These are homologous partners. Okay, it's in 2 twos. This is another one. Okay, this is another one over there. Okay, you have 2 homologous chromosomes, of course, but this, of course, there's 4 in total, 4 number of cells in total. But of course, this is just for you. Is designed for you to understand. Okay, it's not really for a diploid cell. As I said, in humans has 46 chromosomes, so this should actually be 46 chromosomes, but just for to easy up the concepts, to make things easy, really, they just use these 4. Okay, so each 2 is actually a homologous chromosome. Okay, that's what we are saying there. So let's head on then to prophase. Okay, I've allocated 1 next to each phase, because, as we said, guys, we have 2 cell divisions. Meiosis consists of 2 cell divisions. So we'll start with cell division 1. Okay, so that will be IPMET 1, and then the second cell division will be the second, of course, PMET, will be PMET 2. Okay, so we'll start with prophase then. So with prophase, the first thing that occurs is that the nuclear membrane and the nucleolus start to disappear. Okay, I'm sure you guys do know the nuclear membrane. The nuclear membrane, of course, surrounds the nucleus. Okay, so there it is over there, and it has those nuclear pores. And a nucleolus is actually a structure that is inside the nucleus. Okay, it's a structure inside the nucleus that's really, whose function is to help produce ribosomes. Okay, so that's what it really is. It's inside the nucleus, and it helps to uh, produce ribosomes. So those 2 structures disappear: the nuclear membrane and the nucleolus. Okay, that's the first thing that occurs. And then you spindle fibers develop between the centrioles. Okay, so as you can see, there's a lot of terms here. So what are these spindle fibers? Well, if you really take a look at this structure, okay, so take a look at the structure here. It has these fiber-like or wire-like structures, as you can see. So these are what we call the spindle fibers, and these are actually produced by centrioles. Okay, so these are centrioles. So if we would zoom in here, it would be, so zooming in each one of these structures, these spherical structures that produce spindle fibers. So these really look like this inside. So these are actually centrosomes. Okay, And then inside these centrosomes, there's centrioles. Okay? So each centrosome has centrioles. Okay? And centrioles are like these structures that are like 90 degrees like that, and they are really responsible for that production of spindle fibers. Okay, this will help us in the next phases. So we're saying that spindle fibers develop between the centrioles, okay, and then chromosomes are arranged as homologous chromosomes. Okay, so what happens next is the one of the most important events that you are ever going to get in meiosis. Okay, so what happens is that each homologous chromosome. So remember, each when I'm talking about homologous chromosome, I'm talking about a pair, right? As I illustrated. So each pair actually overlaps. It has their chromatids overlapping. Okay, let me just show you what really happens. So it's saying that each one of these, okay, so as you can see, this is a chromatid of this chromosome. So what happens is that it overlaps, okay, overlaps this one like that. Okay, there we go. So as you can see that, let me rather do it again. So again, we're saying that we have this chromosome overlapping that chromosome like that. Okay, so they are overlapping each other, and by doing that, what they're actually doing is that they are actually exchanging genetic material. Remember, each one of these is made up of genes. Yeah? So these are genes, right? So this is made up of genes. Each segment is made up of genes. So when they are touching each other and overlapping over each other, touching each other, they are actually exchanging genetic material. Okay. So what this process we call that the crossing over. Okay, the first important process that occurs and it occurs in prophase 1. Okay, occurs in prophase 1. So adjacent chromatids of homologous chromosomes overlap and touch each other at a point. Yes, almost forget that. They touch each other where? At a point that we call the chiasma. Okay, so the chiasma is essentially the point where they really touch each other. So this would be the chiasma for these two. Um, this would be the chiasma for these two. Okay, use a different color. So this would be the chiasma for these two. Okay, so they are touching each other. Okay, and mind you, each chromosome is different from each other. All right, um, and of course it has different genetic material, and so they are exchanging material, that genetic material, exchanging that DNA there when they are touching each other at a point that we call the chiasma, right? So you have to know all those concepts, guys, all those terminology, right? So we are saying that the chromatid segments, they break off and genetic material is exchanged. So I give you my arm, you give me yours, something like that. Okay. So what this brings, this introduces what we call genetic variation. Remember, we said that these are different. Okay. So we said that these are different at the end. At the end, you get different sex cells or gametes. Why are they different? Well, first of all, um, it was because of the crossing over that which occurs in prophase 1. We'll also learn about this in evolution. This is quite important, guys. Okay, the purpose um, of crossing over is, of course, to introduce genetic variation. Okay, that's why you end up producing children that are slightly different from each other. Okay, and they are slightly different because of this genetic variation, because you have different cells, sex cells, you have different sperm cells, you have different egg cells. Each one of them is different from each other. That's why you're going to um, um, reproduce, of course, offspring that is different from each other, that has different strengths and different weaknesses like that. So it ensures survival as you will see, of course, when we're looking at evolution. Okay, so that is Prophase 1, guys. Very important, very important. I emphasize this crossing over. Understand crossing over. Understand chiasma. That's the point where they exchange genetic material, and understand the reason why. Okay, to bring genetic variation, and this helps with survival of our species. Okay, right. Let's move on to the next phase. So the next phase, remember this is IPMET, so we're done with prophase. So the next phase is M part. So it's the metaphase. Okay, the metaphase, it's really simple. The metaphase and it's the next stage. Really, these are really simple, guys. To really remember the metaphase and the anaphase and the telophase actually. So the metaphase, what happens is that you start to have homologous pairs now that are arranged at the equator. Okay, I need you to look at this first of all. Look at this. Okay, what's happening here? Okay, so at the point where they touched right in the previous phase, which is the prophase. They touched at the chiasma and they exchanged genetic material. Now look at where the points where they touched each other. They have really exchanged. Okay, so this is how we depict that they have exchanged genetic material. So this material has gone to this chromosome. Its genetic material has gone to that chromosome as that, okay, as this homologous pair as well. So once you see this, you see that okay, each chromosome actually has a portion of another chromosome. Then you know that crossing over has already occurred. Prophase 1, we are past prophase 1. So those are the clues that you can use in order to identify a particular phase. So we're saying that in metaphase 1, really, they are not doing anything. They are just arranged. They arrange themselves in the middle, okay, in the equator. And so another thing, metaphase starts with M, M for middle. Okay, so you already know that. Okay, in metaphase, each and every metaphase, whether it's metaphase 1 or metaphase 2, look at that. They are just arranged at the middle, okay, or the equator. Okay, and the way that they are arranged, guys, it's random. They are arranged in a random way. It's what we call the random arrangement. Okay, so look at this. Okay, so these two, they are positioned that way. They could have easily been switched around. Okay, they could have easily been maybe arranged as this one would have been here, this one would have been there. They both of them could have been at the bottom. You know, like the arrangement is random, so um, it really introduces further variation, okay, because you can place any one of these anywhere and there's no way to tell which one is going to be where at which position, so it's a random thing. So this further brings this brings further variation, okay, because we don't know what might happen. This homologous pair could be at the top, that could be at the bottom, they could be switched around. So whenever they ask you what creates genetic variation, it has to be crossing over and also the random arrangement of chromosomes at the equator. Okay, the random arrangement also add or also brings this genetic variation, making of course the resulting in gametes that are not the same. Okay, so random arrangement and crossing over introduces genetic variation. Please hold on to that. Let's go. Okay, so as you can see, guys, we have spindle fibers now that are attached to these homologous pairs, and so of course, as you can see, these are at the middle. So that's the main event that happens there. And then in anaphase, okay, so for anaphase, really the chromosome pairs, they separate, because what happens is that these spindle fibers become shorter and shorter. Okay, so these chromosome pairs, the homologous chromosomes, so each goes to a different, sorry about that, each moves to a different pole. Okay, so this will move that side, this will move this side. Okay, as well as this. So now you are separating the homologous pairs. So you no longer have homologous pairs now, because each homologous partner has went to a different pole. Okay, so as you can see now, you no longer have these together. They went to different poles like that. Okay, so now that's what really is happening in anaphase, is that they actually move to different poles. Okay, each homologous partner of each homologous pair. Okay, so that's really what happens in anaphase. Okay, in metaphase they are arranged at the equator in a random way. In anaphase they separate. That's all. Okay, so and then in telophase, then now that you have each of these chromosomes, individual chromosomes. Now we call them individual chromosomes because they are no longer with their partners. Okay, so now you have individual chromosomes on each side of the pole. Okay, that happened in anaphase. Now in telophase, um, each pole has half the number of chromosomes present in the original cell. Half. What do you mean? The original cell, there we go. There's our original cell. How many chromosomes did it have? It had 4 chromosomes in total. Now, how many chromosomes do we have in each cell? Okay, this will become a cell, that will become a cell. So now you have 2 chromosomes in each of these, each pole. So that's half, right? That's half the number of chromosomes when we compare it, of course, to the original cell. So the cell membrane constricts and divides in half to form 2 cells. Okay, it then constricts and divides in half to form 2 cells, and that is what we call cytokinesis, where you have where you have the cytoplasm separating in half. So the constricting part, guys, I'm sure you guys do eat bubble gum, right? So if you have maybe a bubble gum like that, you could maybe pinch it here, pinch it on both sides. It would look something like that, right? Something like that. Okay, so when you pinch it, like so, that's what constriction is. That's how you constrict it. So now you have this single cell. It has actually divided in half, and it has formed 2 cells. Okay, so it divided into 2 cells. Now each of these cells has half the number of chromosomes, first of all, and it has individual chromosomes, of course, that are not homologous partners. Okay, so that is what is happening. That is telophase. That is the last um, phase of the first meiotic division. Let's head on then to uh, the second cell division. Remember, guys, now you have 2 cells, right? Each cell has 2 or half the number of chromosomes compared to the original cell. Okay, right. Let's head on to the second one. So this is meiosis II. Okay, meiosis II. We don't start with interphase. We start with prophase. Remember, interphase. We said that that is the preparation. Okay, so with Prophase 2, you start up with these 2 cells, okay, that have already separated from, that have already, that were formed as a result, of course, of Meiosis I. So 2 cells. Now, what's different when we compare Prophase 1 and Prophase 2? With Prophase 1, we had homologous chromosomes, right? There we go. We had homologous chromosomes, but now these homologous chromosomes separated. Now we no longer have homologous chromosomes, we have individual chromosomes. So chromosomes are not in pairs. That's what's different. That's how you note that, okay, this is Prophase 1, no, this is Prophase 2. Look at the the chromosomes. Are they homologous chromosomes? What's happening? Okay. So the nucleolus again and the nuclear membrane disappeared. Okay, I forgot to write that down. They disappear. Okay, and the third thing that happens is that the spindle fibers form. Okay, so that's really what happened in the first prophase, okay, where we formed these spindle fibers and also formed where we had these nucleolus and nuclear membrane disappearing. Okay, and note that these are not in pairs and no crossing over, guys. Okay, there's no crossing over. Crossing over occurs only in prophase 1. Okay, please note that. Okay, it's very important that you know the hints. Okay, so create hints for yourself. And then in metaphase 2, we said that in metaphase 1, these will just be arranged in the middle or at the equator. In metaphase 2, that's the same thing that happens, but now you do not have homologous chromosomes arranged, but you have these individual chromosomes in each cell. They are arranged at the equator, okay, like that. Okay, so these are individual chromosomes. I need you to know that, not homologous chromosomes. So that's what happens in metaphase 2. And then in anaphase 2, then anaphase, remember anaphase, that's where the separating occurs. Metaphase, that's where the standing in the middle occurs. Okay, so the anaphase 2, now you had individual chromosomes, right? So the spindle fibers contract. Okay, we know that in anaphase, that's what happens. And then the centromere of each chromosome splits into two. Centromere, guys, and remember that's what holds together or joins these two chromosomes. Okay, these two, I mean, each these two chromatids of each chromosome. Okay, this is the centromere. So it then splits into two. Okay, and then this whole chromatid move to the other side. This whole chromatid move to the other side like that. So that's what actually happens in anaphase 2. Okay, so centromere of each chromosome splits into two. Now two chromatids, um, two chromatids of each separate and move to opposite poles. So that's really what happens. And as you can see, guys, each chromosome now has separated into individual chromatids. Now, these chromatids, we also call them unreplicated chromosomes, guys. This doesn't mean that this is not a full chromosome. Okay, it's what we call an unreplicated chromosome. Okay, so we also count it as a chromosome. Okay, when we are of course counting the number of chromosomes. Okay, so it's just that it's an unreplicated chromosome. Okay, so don't mistake in that. Okay, so that means that now on this side, okay, on this pole, we have how many chromosomes? We have two chromosomes. They are just unreplicated chromosomes, okay? But they are still two chromosomes. It's two chromosomes here, two chromosomes here, two chromosomes there, okay? So, of course, now that you are going to have to separate these, you will still have half the number of chromosomes as um, the, 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 the original cell. So in telophase 2, which is the last phase of meiosis II, a new nuclear membrane forms around the unreplicated chromosomes at each pole. Okay, so now each pole will have so this should have been, I think, oh, okay, all right. So now you have, so let's say, so for these two, for an example, for this one cell, you separated it, right? Okay, and such that a new nuclear membrane forms here. Okay, a new nuclear membrane forms there. and a new nuclear membrane forms there. Okay, right? So it forms at each pole, okay, like that. So that means that you're going to have a new cell there and a new cell there. Okay, so right over here, let's talk about this. Right over here, should have this chromosome and this chromosome. Okay, so I think, yeah, so should have been also um, the other chromosome here, which is like that. Okay, so it should be like that. Okay, so that's what you should have at each pole. So this cell is this cell. Okay, this one is this one. Okay, this one is this one, that one is that one. Okay, let me just zoom out so that you can see everything clearly like that. Okay, because we really it's really quite difficult to imagine all of this in your mind. So drawings can help you guys understand. Okay, so now each cell consists of how many chromosomes? This consists of two, this consists of two, this consists of two, this consists of two, right? So now we have half the number of uh, chromosomes that we began with. Remember, we had four chromosomes at the start. So now we have half the number of the number of chromosomes, and we said that if we have half the number of chromosomes than the diploid cell and the diploid normal chromosome or body chromosome, then we said that these cells are haploid, okay, because they have half the number of chromosomes. Okay, so these are haploid cells, and we said that we then call these, of course, gametes. So these are your sex cells, these are your sperm cells, these are your egg cells. Okay, so that's what happens um, in your sex organs, meiosis, which produces these gametes, and it ensures that these gametes they have half the number of chromosomes. Remember, let's say you as a male have half number of chromosomes. Remember, we said that with humans we have 46 in total number of chromosomes. So this ensures that the sperm cell, your sperm cell, has half the number of the normal diploid cell, right? Meaning which the sperm cell will have 23 chromosomes, the egg cell will have also 23 chromosomes. So that when they fertilize each other, you will have the normal number of chromosomes that each human has. That means that you're going to become a human because each cell of yours will have 46 chromosomes in total. So this is very important. The halving effect, guys, the reduction, as we said there, okay, the process of reduction division, okay, the process of reduction. The reduction part comes from going from diploid to haploid, okay, ensuring that it has half the number of chromosomes, okay, so that these can fertilize each other and a human is born, okay. Now let's take a look at importance of meiosis. Okay, we've already covered the introduction of genetic variation, where you have random segregation or random arrangement um, in the metaphase and crossing over in the prophase one. Okay, so the importance of meiosis, well, it's really quite clear. Well, first of all, there's two reasons why. There's two points when it comes to importance of meiosis. First one is that we are producing haploid gametes. The key word here is haploid, okay? Haploid gametes. So the halving effect of meiosis, that means from diploid to haploid, it overcomes the doubling effect of fertilization, okay? Thus maintaining a constant number of chromosomes from one generation to the next, okay? So this is the first point. So it ensures that the offspring that is going to be born will have the same number of chromosomes as you, okay. So because if you would have a chromosome that would have maybe a different an individual that has maybe more than the normal number of chromosomes, then there's going to be complications, there's going to be problems. So we have to have the same chromosome number. Okay. And then meiosis also introduces genetic variation through crossing over and random arrangement of chromosomes at the equator. So this ensures survival, guys, as I said, okay, survival of our species. So these are the 2 points of the importance of meiosis. Okay, hope you're with me, guys, okay, because now it's about time that we actually do test out this knowledge that we have, of course, just indulged ourselves in. So let's head on then and look at a couple of activities before our time ends. So let's zoom in there. Right, so okay, so first of all, what do we see here? We have a diagram which represents all the chromosomes in a cell that is undergoing normal cell divisions. Okay, it represents all the chromosomes in the cell. Okay, so that means we have a total of 6 chromosomes, right? Okay, cool. So that means that if we have a total of 6 chromosomes in each in this cell, that means at the end we'll produce cells that have a total of 3 chromosomes, right? Yes, half the number of chromosomes. Okay, so another thing that I'm seeing here, guys, is that these chromosomes are actually arranged in pairs. Okay, they are arranged in pairs. Um, so this really means that this should be prophase, right? And prophase one. Okay. Okay. So let's head on then and take a look at the question. So we have 2.1. So 2.1 says name. So we are naming. So it says name A. So 2.1a says name the type of cell division that is occurring in the cell in the diagram. Okay, the type of cell division, guys, it has to be meiosis. Okay. We don't see crossing over in mitosis. Okay, and then B, phase of cell division during which the chromosomes behave as shown in the diagram. So that phase, it has to be prophase one. They're touching each other, guys. We see evidence um, of crossing over. Okay, they're touching each other at the chiasm. If you said prophase, you are incorrect. Okay, remember it has to be specific which prophase. Okay, because in prophase 2, crossing over doesn't occur. And then C, oh, they don't have C actually. So 2.2, 2.2 says where in the human female body would the type of cell division named in question 2.1a take place? Well, in females it occurs in the ovaries, right? In the ovaries, guys. That's where meiosis occurs. Then 2.3 says give the letter and name of the structure that attaches to the spindle fibers. So the letter and name of the structure that attaches to the spindle fibers, that is your centromere, right? So the C. Okay, so C is the centromere. Centromere is um, the place of attachment for spindle fibers. Okay, so we're going to say C, centromere. Okay, read the question carefully because they said give the letter and name. Okay, careful when you're reading questions, guys. Next question, 2.4. How many chromosomes will be found in each daughter cell at the end of this cell division? That's 3. Okay, it has to be 3. Okay, and then 2.5 says describe the events of anaphase 2. Okay, remember anaphase 2, guys, that is where individual chromosomes. Okay, so we can maybe write that down, or maybe let me just for time's sake. So individual chromosomes actually move to opposite poles. So that is what is happening in anaphase 2. So where the first thing that occurs, of course, is the spindle fibers contract, and then the centromere of each chromosome splits into two, and then of course these two separate and move to opposite poles. Okay. So again, it's quite important in life sciences that you do not rush. Okay, write down everything, of course, and ensure that you've written all the necessary points. Okay, so that is what happens. Okay, we are describing there. Okay, right. So that is it for question 2. Let's head on to another question. Let me know down in the comments, guys. Do you find meiosis one of the most difficult concepts uh, to understand or not? And if you have any um any points that you can give us, maybe to so that we can memorize these phases, yes, let us know in the comments, guys. We are here to help each other out. So what do we see here in question 3? The diagrams below represent different phases of meiosis in an organism. Okay, okay, all right. Okay, so with this diagram 1, okay, diagram 1 looks like we have individual chromosomes, yeah? Okay. Diagram 2, these are at the middle, so this has to be a metaphase somehow. Diagram 3, these have, this is anaphase, should be anaphase, because the spindle fibers are shortening, and it should be anaphase 2 actually, really, because you have these chromatids or these unreplicated chromosomes. Wow. Okay. So for 3.1, they say identify part A. What is part A, guys? There's our part A, and it's this structure that joins two chromatids of a chromosome. What do we call that? Call that the centromere, right? And then B, actually have to identify B. So B, we call it so these are two, of course, chromosomes that are arranged in pairs. One inherited from the father and the other from the mother, which have the same genes at the same locus. So of course this is a homologous pair, right? Let's move on to the next one. C. So C, we are supposed to, yes, identify C. Well, C, these are spindle fibers. So that's why it's important that you know each and every single term that you come across, because you can be asked anything. Identify, so 3.2 says identifies phase represented in diagram 3. This phase, this is an anaphase phase, but it's actually the second anaphase 2, because as you can see, we see unreplicated chromosomes. Yeah. Then 3.3 says write down the numbers of the diagrams to show the sequence in which the phases occur. Okay, so told you about the sequence of events, guys. So what happens first? Diagram A, these is well, these are not homologous chromosomes, so this should be prophase 1, really. Yeah, this should be prophase 1, right? Not prophase 1, prophase 2. Okay, remember they should be in these pairs. So this is, we know that this phase is from meiosis 1, and these two are from meiosis 2. So this is so we're going with, so we're writing down the numbers, right? So we start with 2. 2 is metaphase 1, guys. Okay, 1. So we start with 2, and then 1 is prophase 2, right? Right? Yes, yes. 1 is prophase 2. So that's the next one, and then 3 is prophase, not prophase, but rather anaphase 2. Okay, so that's the order there. Okay, set on to the last one, 3.4 says state one difference between metaphase 1 and metaphase 2. Okay, in metaphase 1, um, chromosomes are arranged in pairs. Okay, or we can say that they are arranged, okay, at the equator, um, with their homologous partners. Okay, so yeah, yeah, and of course in metaphase 2 they are arranged um, as individual chromosomes. Okay, yeah. So yeah, let's actually write that down. Let's actually write that down. Just thinking of time. Um, I'm sure you guys do mind a long video. So in metaphase 2, they are individual chromosomes. Okay. You can also tabulate somewhere there. Okay. So tabulating makes things easy. Okay. Set up a table there. Metaphase 1 on the left, column metaphase 2 on the right column. And yeah. Last question. Last activity. The diagram below represents a phase of meiosis. Okay, so this is a phase of meiosis. All right. Okay, what do we see here, guys? Okay, so we already see that. Okay, prophase has actually occurred. I think this is one cell, guys, but it's just that it has constricted there. Okay, this diagram is not the best diagram, really. If this is what I think it is, can't be the best because it looks like 2 cells. But okay, that's fine. This is after anaphase 1. So, well, this is telophase 1. Okay, so it's after anaphase 1. So this is telophase 1, where, of course, the cell constricts. Okay, it's telophase 1. And then 4.2 says draw a diagram to show only the 2 gametes formed from cell A. 2 gametes that are formed from cell A. Well, let's take a look at this. So this will carry on and separate. Okay, only the 2 gametes that are formed. So these two, let's think about this. So these two will have so each one of these will have its chromatids moving to the opposite ends. Okay, So this will move to the other end, this will move to the other end. So that means that the cell that will form from the North Pole will have chromosome that of course is dark in color, okay, and a chromosome that, okay, chromosome, unreplicated chromosome that has this white part, okay, right here. Okay, so this will make up the first cell. Okay, so that's the first cell. And then the second cell, oh man, that's a bad drawing. Okay, so we'll have this chromosome right over here, this one, that will have this white part right over here, and this one is actually all white. Okay, so you really have to imagine this, guys. You can even maybe start from your Prophase 1, Prophase 2, I mean, um, go on and on and on. And this is your cell 2. So that's how it should look, right? And then 4.3, tabulate two differences between Prophase 1 and Prophase 2. When they say tabulate, tabulate. So, well, first of all, what we know is that Prophase 1 crossing over occurs. Okay, so I'll just write P1, guys. Okay, crossing over occurs. Okay, but for Prophase 2, crossing over does not occur. Then, second thing is that you have homologous chromosomes, and then of course for Prophase 2 you actually have individual chromosomes. Okay, so that should be it, guys. Okay, so again, guys, thank you for tuning in. There's a lot more that we are going to do. I need more interaction. Again, I need more interaction. And thank you for sharing this YouTube channel to your friends and family. And I would like you to tune in again. And I'll be producing and releasing a lot of videos each week. So it should be at least one video of each subject, okay, on each week. Okay. So thank you for watching this, guys. I'll see you, of course, at the next video.